Learners, we shall now study about luteolin. Luteolin 3,4,5,7 tetrahydroxyflavone is an important flavone which is naturally found in several plant species. Chemically, it has a C6C3C6 structure and contains 2 benzene rings and 1 oxygen containing ring with a C2C3 carbon double bond. Structurally speaking, structural activity studies have shown that the presence of hydroxyl moieties at carbon 5, 7, 3 and 4 positions of the luteolin structure and the presence of the 2,3 double bond are responsible for its multiple pharmacological effects. We shall now see the sources of luteolin. Luteolin and its glycosides are widely distributed in the plant kingdoms and have been found in many edible plants. Luteolin, which is naturally found as a glycosylated form, is present in different fruits and vegetables including celery, parsley, broccoli, onion leaves, carrots, peppers, cabbages, apples, skins and chrysanthemum flowers. Metabolism of luteolin. As other flavonoids, luteolin is often found in plant materials in the form of glycosides, which are eventually metabolized by intestinal bacteria, cleaved and glucurinated during uptake in the gut and metabolized in the organism. Glucuronidation mediated by UDP glucuronyl transferases and methylation mediated by catechol O methyl transferases are two important metabolic pathways of luteolin in animal and humans. Now we shall look into the health benefits of luteolin. Numerous preclinical studies have shown that luteolin possesses a wide range of biological activities. Although the oral bioavailability of luteolin and its glycosides is not too high, animal experiments have shown that luteolin exerts its biological properties in vivo. Luteolin possesses anti-inflammatory, anti-cancer and antioxidant effects. We shall see the anti-inflammatory property. Luteolin has strong anti-inflammatory activity in vitro and in vivo. Some of its derivatives such as luteolin 7O glucoside have also shown anti-inflammatory activity. The action mechanism of luteolin varies, but SRC in the nuclear factor Kb pathway, MAPK in the activator protein 1 pathway and SOCS3 in the signal transducer and activator of transcription 3 pathway are its major target transcription factors. A clinical trial with a formulation containing luteolin showed excellent therapeutic effect against inflammation associated diseases. Next, we shall see the anti-carcinogenic properties of luteolin. Accumulating evidence suggests that luteolin could be developed as a cancer chemopreventive agent and be useful in cancer therapy to sensitize tumor cells to the cytotoxic effect of some chemotherapeutic drugs. Increasing evidence suggested that luteolin exerted anti-tumor activities in several cancers. Luteolin, it is often associated with its ability to induce apoptosis, which involves redox regulation, DNA damage, protein kinases, which inhibit the proliferation of cancer cells and suppression of metastasis and angiogenesis. Notably, luteolin sensitizes a variety of cancer cells to therapeutically induce cytotoxicity through suppression of cellular pathways and inducing apoptosis pathways. These findings suggest that luteolin can be used as a potential molecule for treatment of different types of cancer diseases. Next is protection from carcinogenic agents. Luteolin can strongly absorb ultraviolet light and derivatives may be used in sun protection products for prevention of skin cancer and early skin aging. We shall now see the antioxidant property. Luteolin also possesses excellent antioxidant properties. Multiple mechanisms may contribute to luteolin's antioxidant effect. Firstly, luteolin functions as a ROS scavenger through its own antioxidant nature. Secondly, luteolin suppresses oxygen radical formation by inhibiting xanthine oxidase activity, which is a ROS generating oxidases. Thirdly, luteolin may exert its antioxidant effect by protecting or enhancing endogenous antioxidants such as glutathione S transferase, glutathione reductase, superoxide dismutase and catalase. Luteolin suppresses lipooxygenase, cyclooxygenase, which are enzymes that catalyze oxidation of the cellular components. Finally, luteolin may chelate transition metal ions responsible for the generation of ROS and therefore inhibit lipoxygenase reaction or suppress non-transition metal dependent oxidation. Learners, to conclude this session, luteolin 3,4,5,7 tetrahydroxyflavone belongs to a group of compounds that occur naturally and called as flavonoids. Plants and fruits rich in luteolin are celery, parsley, broccoli, onion leaves, carrots, peppers, cabbages, apple skins and chrysanthemum flowers. Luteolin is found in plant materials, often in the form of glycosides, which are usually metabolized by the intestinal bacteria, cleaved and glucurinated during uptake in the gut and metabolized in the organism. When it comes to the pharmacological activities of luteolin, it is often associated with its ability to induce apoptosis, which involves redox regulation, DNA damage, protein kinase, which inhibit cell proliferation of cancer cells and suppression of metastasis and angiogenesis. Luteolin are anticipated to have therapeutic advantages and are emerging as a new alternative for cancer treatment.